Now we're going to go into the chapter number 5. In this chapter, we're going to talk about human chromosomes and chromosome behavior. Highly recommended that you go and read this from the textbook before or after watching this video lecture. So here we're going to divide this lecture into three main sections. First, I will go and give an introduction about chromosome and karyotypes. I already talked a little bit about that. In this time, I'm going to focus more on some of the techniques. Then I will talk about the X inactivation and give some examples that complement the video that we watch about with the TED education. And then I will show you some videos about X chromosomes and the evolution of about the Y chromosomes and how they may be in humans, they will disappear eventually. Okay, so let's talk about human chromosomes. So we all know that the human contains 46 chromosomes, including 22 pairs of homologous chromosomes that are autosomes and two sex chromosomes. So here are the same picture, but the difference here is that in one, we organize the chromosomes. So one is A, all of them are disorganized. In B, all of them are organized, and this is what you call a karyotype when you organize mainly by length in the beginning. So this one will be much longer. Well, these ones, the 22, will be very small. And as I said before, from the chromosome number 1 to the chromosome number 22, they are all autosome chromosomes, the only sex chromosome. It will be the X and Y chromosomes. And one of the most common techniques used is called fluorescent in situ hybridization, or FISH. You can use this for the abbreviated version, and here is what you will see: just labels that are fluorescent. And what is actually happening? What is actually happening is that if there is a chromosome there will be DNA, and when you heat up the DNA, you will break the hydrogen bonds, and now you will find the version of the DNA denatured. And so now you will have, instead of two strands, you will have one. Then you add a probe that will be fluorescent. So here the star represent the fluorescent labels of the probe, that it will be complementary to one of the strands. So if you want to label a particular section of the DNA, what you're going to do is to add the probe, and later, if there is complementary, they will attach to each other, and you will see sections of the DNA of the chromosomes that they will label, and the other ones that they didn't label, they will be washed during the process, so they will be removed, and only the sections that will be fluorescent, it will show you that that sequence is there. Okay, so to summarize, FISH is a molecular cytogenetic technique, and what it uses a fluorescent probe like this one, that what it does, it only binds to particular parts of the DNA. And uh, these type of techniques, these ones, uh, um, when you use fluorescent, this one, antibody stainings, and you will hear a lot of those ones. There are many. What they are doing is a process which vividly paints the chromosome or portion of the chromosomes. And possibly one of the most common that you will hear the most is this one, DAPI. And DAPI label the nucleus. And all of these ones use fluorescent probes that bind to the chromosome with a high degree of complementarity. Why you have to high degree of complementarity? So you don't have attachment in regions that are not specific, that it could happen. So you always have to check to have a positive and negative control to know that your probes are working very well. So now that we know how these type of stainings are done and how they organize it, let's talk a bit again about this. and about its importance. So here we talk about that this here in V is a karyotype, and a karyotype is when you stain and photograph a preparation of metaphase chromosomes and arrange them accordingly to the following parameters. The size, as I showed before, when we compare 1 and the 22, if you see the sizes start getting smaller, and the centromere, the location of the center of the chromosome, that later we will talk in a second to remember that concept. And, and this technique is very useful, and I'm going to show you in a second how it will allow us to see complex chromosome rearrangement. And those complex rearrangement, you will see that play a key role during evolution and also during disease like cancer. Okay, so this will allow to do this very fast and in a very cheap way. Okay, before moving into more explanations, I just want to give you a quick reminder of the different sections of the chromosome that you need to remember. So centromere, that is here, that is the point where sister chromatids are joined together. So here you have one chromatid, and here you have the other chromatid, and they are joined here by the centromere. One common mistake is to confuse centromere with centrosome. Yeah, that is a common mistake. Centrosome is something different that is involved in the cell division. Okay, centrosome is different from centromere. Okay, so this is not a centrosome. Okay, the P is the short arm upwards. Yeah, and this P, because it comes from French, that is petit, is supposed to be the short arm. Yeah, this Q also from French, that it means the tail, the long version of a chromosome. And telomeres are, as we talked before, is the ending section of the tips of the chromosome in both sections of the chromosomes. And again, each one of these is a chromatid. Yeah, here you have one chromatid, and here you have the other chromatid that are joined by the centromere. Okay, now that we have those concepts clear, let's move into the next section. Okay, here we have a real chromosome, and again, all the same parts of the chromosome that we just talked: telomere, short arm, long arm, and again the centromere. However, if you see this chromosome, you can see that there is light bands, and I want that you pay attention, that you remember that. And you also have dark bands, and this is a type of staining, again, that is called a G banding or GMC banding, based on the name of the staining. In the light bands, you will find that those sections of the chromosome replicate early in the S or synthesis phase. They are less condensed chromatin, and for that reason, as they are not as packed, they are transcriptionally active, and also they have their sections. They have more repetitions or of G and C. On the other hand, the dark bands, they replicate late. They are more condensed chromatin, and they are rich on adenine and thymine. Okay, so these two regions are important to talk about it and to differentiate it. So again, this is the type of staining that I was talking in the previous slide, the G banding, and here you can see a schematic of what you will observe. Again, this is another technique of in cytogenetics that is much cheaper than the molecular biology, and is still useful. It's used to stain condensed chromosomes, so chromosomes during metaphase. And if you see, you can see interband regions based on the patterns of the stainings, and those interband regions between light and dark regions are very well characterized, and there are some familiar genetic landmarks. So, for example, you can determine the blood group. You can determine with this the color blindness and different type of diseases. So this is an easy way and a cheap way to detect several type of diseases. Here you can see an schematic from your textbook that show how these banding patterns are described in humans, and you can see different letters here: A, B, C, etc. And those are grouping the chromosomes by size, as we previously said. And again, those patterns are not only studied in humans, in, for example, in Drosophila. The same type of information you can find it online describing all these cytogenetic studies. Okay, so now let's talk about another way of to classify the chromosomes. So it's not by size, but the location of the centromeres. So first here you can see the metacentric, and if you see this schematic here, the centromere is located in the middle. Now let's go to the second one, the submetrocentric, that instead of being in the middle, the centromere is located closer to the end, not exactly in the middle. The third type that is shown here is the acrocentric, that is really close to the end. And the fourth one, the telocentric, is the one that is close to the telomere. Yeah, that is just at the end. And so remember, in this slide, I talk about chromosomes and that they are classified also relative to the position of their centromere. So, for example, we have the metrocentric that are in the middle. I also talk about the submetrocentric that are close to the end of one of the chromosomes, the acrocentric that is really close to the end of the chromosome, and of course the telocentric. And something very important to tell you is that when chromosomes has no centromere, or they have two centromeres or more, they are genetically unstable and they are not able to go through mitosis normally, and they are these are an indication that something wrong is happening with the cell division, and that those cells, or they should go through apoptosis, or they are carcinogen, or they are cancerous cells. In this schematic, I'm going to also talk about these linear monocentric chromosomes and how they look during cell division. So, for example, the metacentric during cell division, they will have a V shape, while the submetrocentrics, they will form a J shape, while the acrocentric during cell division will form more like an I shape. So again, remember, if they have equal arms during cell division, they will the metacentric will look like a V, while the submetrocentric will form a J shape, and the acrocentric will form an will look like an I. They will have an I shape. And why this is important? Because again, all these are help us to identify the chromosomes and detect abnormalities, and this was the only way before we were able to sequence and to try to understand each one of the genes. It was the only way to identify abnormalities in just from a very macroscopic perspective.